química, analisando o fluxo de informação do DNA para o RNA para as proteínas na nome.
Hey there, I'm Josh, and welcome to the Chem 101 series. In this series, we're going to go over an array of topics, including the central dogma, as well as other concepts well known to chemists and other participants here in the field of drug discovery. But first, let's look at the central dogma. Overall, the central dogma is the explanation of how we get all the different macromolecules within the cell. And the first macromolecule we're going to look at today is DNA. Now, I'll go ahead and pull it up for us right here. And as you can see, this wonderful geometry that's formed here is able to store information along its basis. And we'll go more into detail about how it does that in the next tutorial. But something to note about the DNA sequence is that it is the blueprint for so many molecules within the cell, and it's very, very important that the copies are stored in the nucleus and that they're kept perfectly in the same sequence as when they started. This way, as cells replicate and as life goes on, these sequences are not corrupted, ensuring that cell replication can move forward properly. Now, mutations are when we change or lose spaces within the DNA chain, and this can result in all sorts of disease. So, since DNA is so important, instead of just taking the DNA and making molecules from it, we instead undergo a process known as transcription. Now, transcription is the process of DNA being converted to another readable format, and that's called RNA. Transcription is exactly what it sounds like. We take information stored in the DNA, copy it as a template, and now we have a new copy, which we call RNA. In this case, messenger RNA. Now, messenger RNA keeps the same sequence from DNA, but has a slightly different structure and slightly different features. The R in RNA, for example, simply stands for riboxy, versus the D in DNA, which stands for deoxy. So now we're on the same page. There are slight structural differences between DNA, RNA. What's more important are these functional differences. RNA is able to be used outside of the nucleus in order to do a host of tasks, including, but not limited to, the information it stores. But it also can form secondary structures such as tRNA, mRNA, which allow to perform other tasks. Now, we're going to be looking at what mRNA does, where it acts as a template to give us instructions that can be translated into amino acids, which will make up a protein. Now, any changes that occur back in the DNA, therefore, can eventually become changes in the protein, which is why it's so important that DNA sequences are preserved. RNA can also be changed, and there are other ways that RNA can be affected that have downstream effects on the protein that it yields. Proteins do so much more, though, than just provide information. They can do just about anything within the cell. They can provide structure, and they can act as motor proteins to move things about the cell. Really, we cannot emphasize enough how important each of these macromolecules are to the drug development process. Now, depending on your skill level and preliminary knowledge, you may have gotten lost in some of the jargon I've thrown at you, but don't worry. We're going to go over all of these words, all these macromolecules, and all the other vocabulary terms in our next tutorial, Chem 101.2. Hope to see you there.
Bom, é isso aí. Sai do DNA, cria RNA, vai para fora da célula, do núcleo da célula, e cria proteína. E essa proteína forma os nossos tecidos e outras proteínas fazem com que esses tecidos movam-se. Interessante.